Hi, I'm Bob Lessick at Johns Hopkins. In this lecture, we will examine how to input a protein sequence and output three types of predictions. The isoelectric point and molecular weight are very simple to calculate. We will also examine the prediction of where a protein goes within a cell after it is translated or synthesized. We want to first look at exactly what is meant by isoelectric point and molecular weight for a protein, and then go ahead and perform those calculations. We'll shift to targeting or localization. Where in a cell can proteins go? Let's look at some possibilities. Finally, let's try to predict where proteins go based on sequence. Many proteins have short stretches of sequence that serve as tags that molecules involved in transport might recognize. Let's start with molecular weight. Molecular weight is measured in Daltons. The word Daltons is not capitalized, but the abbreviation DA is capitalized. For those of you familiar with chemistry, a Dalton is synonymous with a unified atomic mass unit, which is close to the weight of a single proton or single neutron. The unit is named for John Dalton, an English chemist-physicist known for atomic theory. The molecular weight in Daltons is equivalent to grams per mole, except instead of measuring the mass of a mole of molecules, the Dalton refers to the mass of a single molecule. I should note that molecular weight is technically an incorrect term, since the Dalton is a unit of mass, not a unit of weight. The same is true for grams and kilograms, both units of mass. Weight is measured in the English system with ounces and pounds, primarily in the United States. What's the difference? Weight is affected by gravity and mass is not. Although molecular mass is a more correct term, molecular weight is generally accepted and more commonly used. Isoelectric point is usually abbreviated as PI, a lowercase p and a capital I. That looks a lot like pH, and there's a reason for that. The isoelectric point is the pH at which the protein has no net charge. That is, all of the positively charged atoms are equally balanced by the same quantity of negatively charged atoms. So if the isoelectric point is less than 7, remember 7 is neutral pH, then that protein is acidic. If the pI is greater than 7, then that is a basic protein. To put it another way, acidic proteins have a net negative charge at neutral pH. Meanwhile, basic proteins have a net positive charge at pH 7. These calculations are among the easiest in sequence-based bioinformatics. For molecular weight, each amino acid has its own molecular weight. Add them up, and then subtract the weight of water for each peptide bond in the chain. That's because one H2O molecule is released upon peptide bond formation. Isoelectric point is a little more tricky, but not that difficult. There is a tool called the Compute PI-MW tool, PI for isoelectric point and MW for molecular weight. You input the sequence, and the output contains both the isoelectric point and molecular weight. I should add that these are important measures. There's a reason to do this. In addition to knowing those important parameters, both measures are useful for developing strategies to separate and purify proteins in the lab. Here is the output for human beta globin. The calculated molecular weight is 15,867.22 Daltons. Most would more commonly round that off to 15.9 kilodaltons, or even 16 kilodaltons, meaning 16,000 daltons. The isoelectric point is 6.81, slightly acidic, but very close to neutral. At neutral pH, this protein would have a very slight net negative charge. Now let's transition to protein localization. That is a more difficult prediction, since the signals that are found in proteins are short. Let's examine a few places proteins could go in a eukaryotic cell after translation. The secretory pathway is a mechanism for transporting molecules outside of the cell. Sometimes cells need to release proteins for a variety of possible reasons. Try to think of a few reasons that might be useful. The Wikipedia entry on secretory pathway explains it very well. Proteins that utilize the secretory pathway are deposited into the cell's endoplasmic reticulum as they are being translated. From there, they travel to the Golgi apparatus and then via secretory vesicles to the plasma membrane. Some of those proteins stay in the plasma membrane and some are released to the outside. However, other proteins are destined to stay in the cell. Some function primarily in the cell's cytoplasm. The cytoplasm is an aqueous region that is basically everything but the organelles of a cell. Here are some of those organelles. The nucleus hosts the genomic DNA. The mitochondria are the energy centers. The peroxisome is where fats are broken down. And the lysosome is a digestive center. These are all places that require specific enzymes and proteins in order to function properly. So getting those proteins to those locations is very important. And in plant or photosynthetic cells only, a possible destination is the chloroplast. There are many prediction programs available on the web. Many are general predictors. Put in an amino acid sequence, and the program predicts its destination. Some lists possibilities from most likely to less likely. Some programs are specific for a single organelle. For example, a program called mitopred, well, you might not be surprised that predicts whether or not a protein goes to the mitochondria. It's a yes-no question, and the output of such programs is usually pretty simple and expressed as a percentage. For example, the prediction might say that a protein is 78% likely to be mitochondrial based on its sequence. Here are just some of those web servers, and there are quite a few out there. Let's do an exercise. First, find the yeast PEX8P protein at NCBI. The accession number is NP_011591. And let's get that protein sequence in FASTA format. Then go to these servers and enter the protein sequence. Make sure it is set for eukaryotic prediction if that is an option. And we'll see what we get from Predator, Cello, and Loketree. This is Cello. I believe it's pronounced Cello because it's a cell prediction. But it's a Taiwanese-based program and web server, so I'm not positive. Maybe it is Cello. There are two important things to do before running this program. First, switch to eukaryotes. The default is set for gram-negative bacteria. Gram-negative bacteria have no organelles. So that prediction is very different, mainly secretory or retained in the cell. Then note that there is a protein sequence already there. That's an example sequence that is there by default so that users can see how it works. Be sure to delete that sequence before you enter your sequence. Cello offers 5 different parameters to make its prediction. The amino acid composition, the N-terminus, and the physicochemical properties suggest that this protein goes to the peroxisome. Based on examination of the partition sequence and neighboring sequences, it appears to be a plasma membrane protein. Cello is an example of a jury prediction strategy. The consensus of the jury, based on the results of the 5 different predictions, suggests strongly that this protein goes to the peroxisome. Do a web search for PEX proteins, P-E-X proteins. That will give you a good idea of where this protein actually goes. So to wrap it up, we know that we can enter a protein sequence and get a reliable calculation of the isoelectric point and molecular weight. And that many localization predictors are out there. Some are general, some are specific for certain organelles. Give some of them a try. Good luck.